Thank you for joining me for this brief video presentation of our poster on the screen of natural products to stimulate lubricin production in human cartilage. So, as a brief introduction, lubricin is a potential target for disease-modifying treatment of osteoarthritis, and it's those cells at the surface of the articular cartilage that are secreting that lubricin. We went with a natural product library because it's a good source of bioactive molecules, a 3D culture system with human chondrocytes, and with a secreted gaussia luciferase reporter cell line. So what we did was take a standard aggregate culture and to make sure that our transfected cells were actually still chondrogenic, primary human chondrocytes. We formatted them from the synovial matrix into these non-adherent U-bottom plates and cultured them for over 3 weeks. Over those 3 weeks, we were able to do a temporal analysis of the media. And looking at the TGF-beta dose response curve here with TGF-beta on the x-axis and the lubricin promoter-driven luciferase on the y-axis, we can see a nice dose response, both at day 10 and at day 22. Now, because lubricin is a secreted molecule, we're actually able to take that media and look at the media for the lubricin content by ELISA. So we can see that there's a nice dose response by ELISA too. And just to make triply sure that we're getting a correlative response of the luciferase with the lubricin, we did the gene expression and the normalized gene expression for the Gaussian luciferase against the lubricin showed a really good correlation. In terms of the histology, we can see that we've still got GAG staining, so the GAG in the pink is increasing with increasing TGF-beta dose. Same for lubricin. And, because we're looking at articular chondrocytes, we want to make sure that we've got that chondrogenic phenotype using that type 2 collagen staining. So, we've shown that these cells are still chondrogenic, and if you want to see more on their utility, go see the bioprinting poster at 597. So now that we've established that these cells can report on lubricin and that they're chondrogenic, we screened this natural product library. Took 390 compounds and tested them from all the way through 3-week culture with sampling on Wednesdays and Fridays for luciferase. At the end of that screen, what we assessed was the metabolic activity, and then we formally fixed those pellets and embedded them for histology. So through this experiment, we can see that there's this temporal response that in the DMSO control here in red, there were actually quite a few compounds that were stimulatory, especially at this day 10 point. And when we looked at that statistically, we can see that these 3 compounds really stood out. Okay, and so looking at the compounds and their effects overall, we can see that most of them were inhibitory, both at day 10 and at day 22, with some just not having really any effect. But then there's this small portion of compounds that had a stimulatory effect on the lubricin content. If we then kind of narrow that down by the compounds that were not having a negative effect on metabolic activity, we can see that there are these 9 compounds that still stood out as being stimulatory to lubricin production. Looking at the 3 identified compounds at day 10, we see that all of them actually seem to decrease the metabolic activity. Interestingly, they all increased the aggregate size, but in contrast to the DMSO control, which increased from day 10 to day 22, they all actually lost aggregate size over that time period. So looking at the kind of compounds of interest, compound 270 actually didn't find any analogs or predicted targets with this in silico screen. Compounds 188 and 217 had very similar potential targets, with compound 217 having lots of kinase activity or potential kinase activity. So, in summary, we have developed the primary human chondrocytes with a lubricin reporter. The lubricin reporter works, they don't lose their chondrogenic activity, and we successfully screened a natural product library. Many of the products that we looked at were actually decreasing the metabolic activity, but a fraction still stimulated lubricin production. And we have some potential leads to go on for further study. Any questions on this, please feel free to find me at the ORS or contact me by email or over Twitter.